Alright, class, so I'm going to be talking a little bit about the digestive lab that you guys just finished. I guess not just finished, but finished a couple weeks ago. And let's first begin with the first experiment that you guys used using glucose iodine. And glucose iodine, remember we talked about how it's an indicator for complex sugars like starch and causes it the color of it from being brown to being blackish, darkish color if starch is present. So looking at this, your very first experiment here, you have just water with glucose iodine with it. So when you had glucose with water, what was the results that you guys ended up having? It should come out to be kind of like brownish still, giving us a more negative for starch. So their starch is not present in this particular case. So then once again, this is what kind of it is a control experiment because all we're doing is adding water in this particular case. And so that means that here I should use this, which means that we're just adding water with the indicator itself. So there was no other variables that we're adding to this. So this will be our negative control then. Okay, and notice how the second one here, where we add just water, and then we do add some starch, and then glucose in the end. So here we have, basically we're adding our second experiment, we're adding starch. So we're adding starch with glucose iodine. Okay, and remember glucose iodine is an indicator for starch, so it changes this into like a darkish blackish color. So oops, I just erased it, so it's like black. So that means it is a positive reaction, or positive for starch. Of course, it makes sense because we're actually adding starch in this particular case. So making this another control, and this is our positive control because we're purposely adding starch to give us a positive result, while we're purposely adding just water to give us a negative result, and that's why this was a negative control over here. Okay, and then the other test tube that we ended up using, we put some saliva inside of it, add some starch, and then add glucose iodine. So in this particular case, we're adding saliva plus some starch and plus some glucose as well. So now we're adding a variable into it, which is saliva. And the end result, well, a couple of things about this end result. Well, since we only digested it for, 20 minutes or so, or 30 minutes, there will be some starch left over because not all the starch has been digested by which enzyme do we find in saliva? Salivary amylase. Okay, you can probably not read what I just wrote there, but amylase. And remember, amylase breaks up starch, the polysaccharide, into smaller and smaller polysaccharide. So just it doesn't break it all the way down to glucose yet, but it possibly can break it down to maltose, which is 2 glucose together. And so over here, because we have starch in here and not all the starch is being digested by the salivary amylase, that means that there's going to be some starch left over, making it a black color still. due to the glucose being an indicator for starch, and that means that it's still positive for starch. And this is our experimental test tube in this particular case, while these were controls that we saw over here. Alright, so that's that. Going on to the next test tube is an unknown, so each person had their own unknown, so I'm not going to go into that. So that was it for the polysaccharide test. But, okay, I guess one more thing I forgot to mention, but imagine if we ended up allowing this test tube here with saliva, starch, and glucose iodide. We let it sit there for, let's say, 2 weeks or a month or something like that. What do you expect to have at the very end of this long period of time where salivary amylase is constantly digesting the starch? You're going to end up having possibly what kind of color? It'll be kind of brownish in color still. Because at this point in time, it is possible that all that salivary amylase digested all the starch, so you won't have any more starch left over. Most of it is going to be digested down to simple sugars or disaccharides. And at this point in time, there won't be any starch left, so it's brownish in color, so the glucose doesn't detect any starch, making it a negative result for starch. That's after like several days or whatnot type of thing. I've never actually tried it out, but that would be pretty nasty if we had saliva sitting around for that long. Okay, so moving on to the second experiment where we tested monosaccharides using the Benedict's solution. And remember, Benedict's solution test for monosaccharides, it's blue in color, generally speaking, and depending on how much monosaccharides are present, it changes from green to yellowish to orange. Okay, I can't find orange on this, but orangey red kind of thing at the end. So that's what Benedict says. The more concentration of monosaccharides there are, the greater the amount of color change due to the Benedict's solution. So on the very first experiment, we added water and just glucose iodine. Sorry, not glucose iodine, it's Benedict's. So just to give what kind of control, and if you said negative control, you're correct. So we just had Benedict's for the first test tube plus water. So we know water is not a monosaccharide, so this is purposely going to give us a negative result. So it's going to turn out blue, which is negative for monosaccharides. Monosacs. So if it's blue in color is negative, and so and we purposely did not put any monosaccharides in there, so this is going to be our negative control. Okay, so the next one, I guess we did wanted to test this out, but we put in starch. I'll put it over here. Starch with Benedict's, just to see if starch reacts with Benedict's. Starch is once again a polysaccharide, so it's not supposed to going to react with the Benedict's solution, because Benedict's solution is an indicator for simple sugars like monosaccharides and disaccharides. So we're going to still have a blue result, meaning that it's negative for monosaccharides. So this is actually an experimental test tube that we're using once again, because we're actually adding starch, and that's not, well, although I guess it could be a negative control, but technically speaking, water would be the best negative control, since we do know that starch is a carbohydrate, so we don't know for sure whether or not Benedict's solution will react to it. So this was more of an experimental, just to see if it does or does not. And this other one here, we have the saliva mixed with starch, and then we let it incubate for a little bit. So we have saliva plus starch, and then plus Benedict's. And of course, remember, saliva has what kind of enzyme again? Salivary amylase. And what does that do? Where is it found? It's found in the salivary glands in our mouth, oral cavity to be more precise. And then that's going to digest the starch into maltose and eventually to a glucose. And so that glucose will react with Benedict's, and that will change it into, depending on how long we set it in there for incubating with the salivary amylase with the starch. So the longer that we let it sit in there, the more and more glucose that will be produced by digesting the starch. So let's just say it turned green. I know for some of you it ended up turning like orangey color. It just shows you the more amounts of glucose that's present versus the lesser amounts, which is greenish in color. So you might be asking, well, where's our positive control? Well, our positive control, as we can see, we did end up adding a fourth one here. I think we added here, showing you that we added some water, some glucose, and then boiled it. So here we added water and glucose, and of course now we're adding glucose, so we're going to purposely try to have a positive result because we know Benedict's reacts with glucose, so it's going to be really bright in red color since it's pure sugar that are pure glucose that we're actually putting into it. So of course it's going to be positive for monosaccharides. And this of course will be our positive control due to us actually adding glucose in this particular test tube over here. Alright, so that's it for carbohydrates. Let's go on to proteins now. So with proteins, we have the very, I guess we'll talk about the indicator first. The indicator is biuret reagent, and remember, this detects peptide bonds, so it's an indicator for peptides, basically. And the more the amount of peptides, the purplish color that it gets, the less amount of peptides there are, it becomes pinkish in color. So the very first test here, we're going to have a negative control where we're just adding water plus biuret. And since water doesn't have any proteins in it or peptides, it's going to give us a blue color, making a negative result for peptides. And this once again is our negative control. Alright, here we added ovalbumin, which is found in egg whites. It's basically egg white protein. And of course, I just mentioned it, but egg whites have a lot of protein in it. And this ovalbumin, we added some HCl with it, and then added some water with it too. So we had albumin or ovalbumin, whatever, both of them are the same. I know I wrote it in yellow, but it's not the egg yolk, but egg white, plus HCl, plus some biuret. And since we do have albumin in it, that's going to cause it to be positive. I guess if we wanted to be really technical, it should have just been albumin with biurea to be the positive control. Since we did add some HCl, it makes it a variable, causing it to be more of an experimental test tube. But I think the reason why we ended up adding HCl, you'll see in the next test tube that we were talking about. Anyway, albumin does react with biurea. So it's going to give us a very purplish in color, since albumin is huge protein, so it's going to give us a very purple color. So that's going to be a lot of big positive for peptides, since there's multiple peptides in a polypeptide chain, which is a protein, or which could be a protein. Okay, so here in this next one here, we're going to add albumin, some HCl, pepsin, and then the biurea reagent. So this experimental design, we're going to add albumin, some pepsin, and some HCl as well. Of course, with biurea as well to test it. Okay, so doing this, what is pepsin? Pepsin is a protease, so it's an enzyme that breaks down proteins. We just mentioned how egg whites contain a lot of proteins in it. Where is pepsin found? Pepsin is found in the stomach, and so this enzyme works best at low pH, hence the HCl. Remember, HCl is also produced by the stomach as well. So pepsin works best at low pH, which is the HCl, so it will then end up digesting the albumin. So if it digests the albumin is a polypeptide chain, and so if it digests that, it's going to make it into smaller and smaller polypeptide chains, giving us fewer and fewer peptides. And so we're going to expect to have more of a pinkish color, not as purple as the whole albumin that we had earlier, the whole polypeptide chain, since pepsin ends up digesting that polypeptide, making it shorter and shorter. So here we are positive for peptides, but it's not as positive, or not as much as the previous one, the positive control here. This one was our positive control. So this was our experimental one. Okay, our very next one here, we're testing albumin in NaOH with some pepsin. So we have albumin plus pepsin plus NaOH plus biurea. And NaOH is a base. And so what do you think will happen with the pepsin enzyme? It's going to cause it to denature, making it less effective on digesting the albumin. Unfortunately, I think a lot of us, when we tested it out, we weren't able to actually see that. But if that was the case, we would have expected it to be still a bigger polypeptide chain, since pepsin would not be optimally functioning at this time, so it won't digest the albumin as much. So it's going to be very positive for peptides. So it'll be pretty much undigested because the pepsin is not working as well due to the high pH of NaOH. Let me pause this for a sec. Sorry about that. I wanted to actually show you guys a picture of what Jessica took in lab about this particular experiment. However, the picture is not showing up since I get the guess because I don't have Outlook on my computer here. So unfortunately I can't share that with you guys. But let's go onwards with the next experiment here, using the lipid digestion. With the lipid digestion lab, we're utilizing litmus milk. Litmus is purple in color when it's high pH. It's a pH indicator, by the way. Litmus is purple in color at high pH, but at low pH it turns red. And milk, as we know, is slightly basic, so when we add milk to litmus, it's going to be this purplish color. How do we turn milk to be more acidic, though? Well, we're using whole fat milk, so there's a lot of lipids in here. And what kind of lipids are we talking about? Triglycerides. What are triglycerides made out of? It's made out of glycerol and 3 fatty acids. And the acids portion of the fatty acids will cause it to lower in pH because they're acid, making the litmus detect the acidity, changing it into a reddish color. So, if we look at this very first one here, it's adding water with litmus and a drop of HCl. So we have litmus plus water plus 1 drop of HCl. So what is the color going to be after adding all these together? If you said red, you'll be correct. Because we're adding HCl, and HCl is a hydrochloric acid, it's going to be very acidic. So it's going to cause it to be very low in pH. What kind of control is this? Well, we're purposely adding hydrochloric acid to give us a positive result for it changing to acid. So this is our positive control. This next one here, we're just adding water and then litmus milk to it. So we're just adding litmus milk plus water. So what's still going to be the end result? We're going to have it to still be purplish in color. Since we're adding water, we're purposely causing this to have a negative result, or in this case, a positive pH result. So this is our negative control. Okay, in our final experimental test tube here, we're adding pancreatin with litmus milk. So we add pancreatin, which by the way has what? It has all kinds of enzyme. We have both pancreatic amylase, we have trypsin, chymotrypsin, and carboxypeptidase, whatever. All 3 of these are proteases, and finally we have pancreatic amylase. My handwriting is getting messier and messier. What am I talking about? Not amylase, but lipase. And what does lipase do? It's going to break triglycerides into glycerol and fatty acids. So we have pancreatin added to the litmus milk. And of course, the pancreatic lipase is going to digest the milk triglycerides, the whole fat milk triglycerides, into glycerol and 3 fatty acids. And that 3 fatty acids is going to cause the litmus to change color from purple to reddish in color due to the lower in pH due to the fatty acids. So we compared that with our positive control and see, yes, it does change kind of pinkish, reddish in color, giving us our positive result. Finally, we have the Sudan 4 test about lipids, or triglycerides in particular. So what happens is that in this very first one, we're adding water with Sudan 4. Sudan 4, once again, is a fat indicator, a triglyceride indicator. So it's going to be, it is a hydrophobic indicator, so it's going to be attracted to triglycerides, so it's going to kind of stick to it. So in water, so the very first test tube, we have water plus some Sudan-4. Sudan-4 is reddish in color, so you're going to see that the ones that have triglycerides in them will stick, it will cause that to change reddish in color. Even though we're adding Sudan in water, it's going to cause the water to be reddish in color as well. But you'll notice that when we looked at this, it was very transparent, translucent, so we could actually see through it. So versus when we actually had vegetable oil in it and Sudan-4. So the next experiment, we have vegetable oil with Sudan-4. And we notice in this particular case, there was an oily layer on top due to the vegetable oil and water in the bottom. And when we added the Sudan-4, even though there was reddish in the water portion, most of them concentrated in the vegetable oil layer. And plus, it was a lot darker in color in the vegetable oil layer there. So this was our positive control. Well, this was our negative control here. Okay, negative control. Negative control like that. Okay, and this final one was our unknown test, experimental test, just to see whether or not it's going to be positive for the Sudan-4 test or negative. So that's how you will test for triglycerides. A simpler way is using a paper towel test, which we did not do, but I think it's pretty commonplace if you think about it. If we use a paper towel, we add a drop of water on this paper towel here and a drop of oil over here and let it dry for a little bit. What do you expect the water portion will look after we let it dry for 10 minutes? It's going to dry out. It will leave a little water spot, but it's going to be dried out. Versus the oil spot here, it's going to still stay wet even though we let it dry for 10 minutes. It's going to still look like it's a little wet. So that's another way of testing your unknown. If you had an unknown and used the paper towel test, you put a drop on it, let it stay there. You might as well put a water drop next to it and an oil drop next to it. So this is our unknown. This is our oil. And this is our water. And then you let it stay there for 10-15 minutes and see what it looks like. If it looked more like the water spot, then the unknown most likely does not have triglycerides in it. If it looked more like the oil spot, then we know that yes, it does have triglycerides in it. So that was another way of doing it, although we did not do that in lab. So that's it. I just wanted to kind of go over the lab and our expected results from this lab, and hopefully you were able to gain something from this video lecture. Alright, that's it. See ya.